the last class of macromolecules are the nucleic acids. So the proteins were Roman numeral number 4, so this is going to be Roman numeral number 5 is nucleic acids. Okay, so let's talk about what a nucleic acid is. All right, so nucleic acids are polymers. Remember what a polymer is? Polymer means, remember, it's a long chain of a smaller molecule. Are polymers called polynucleotides. What does that mean? Whoops, I'm off the screen. Poly means many, and nucleotide means a nucleotide. So let's talk about what that means. What is a nucleotide? So the monomer of a nucleic acid is a nucleotide. So let's talk about what a nucleotide is. It consists of, and I'm going to put a colon here. All right, so let's look at what it has. It has a nitrogenous base. That means it contains nitrogen. It's a base that contains nitrogen. What is a base? And remember, it increases pH on the pH scale. A pentose sugar, let's see if we can make that capital, a pentose sugar. All right, ose means it's a sugar, pent prefix means 5. So what this means is it's a 5-carbon sugar. And let me give you an example here of this. It could be ribose in RNA, and we'll talk more about this later, and deoxyribose in DNA. And then the last thing that's part of a nucleotide is a phosphate group. So let's draw what this looks like. All right, so you have a phosphate group. Let's do that first. And I'm gonna color code. All right, let's do purple, green, and orange. All right, so let's do purple as the phosphate group. So I'm gonna draw phosphate group purple. So I'm not gonna draw every single atom. So you have a phosphate group, attached then, bonded covalently, and it kind of goes like this. And then you have your pentose sugar. So pentose, it's gonna look like a house with the O at the tip, and then like this, like that. All right, so that's your sugar. And then let's do your base. Your base, it's gonna come out here. And let's say your base, let's do your base in orange. All right, so let's say it's like this. All right, hexagon there, and that's your sugar. Phosphate sugar base. So that is, so your sugar is black and your base is orange. That's your nucleotide. Every single nucleotide has those 3 components. What changes is if the nucleotide is making up RNA or DNA, which are your 2 kinds of nucleotides. All right, so as we wrote here, the sugar can change. It can be ribose or deoxyribose, so that can change. And then also, this is supposed to be your base, not your sugar. Here, I just realized I put an S, a B. That's supposed to be your base. your base can change. All right, so let's talk about the types of bases here. So I'm gonna go back to my black pen, all right, and look at this. So the base, it's right here for number 2. The nitrogenous, I'm just going to put N bases instead of writing the word nitrogenous. You can write nitrogenous if you think it's going to be confusing to you. Nitrogenous bases can be single or double ringed. And I'm going to put here C figure 327. I'm going to show you that in just a second here. All right, so I just showed you here a single ring for your base here. That's one type of base. You could also have a nucleotide, phosphate, and then the sugar. And then you could have your base be 2 rings, all right? So you might have it like this, and like, see how I mean by 2 rings there? All right, that would be a double ring. So these are both examples of nucleotides, but what's changing between them is the bases. So let's write here, you can have single ringed nucleotides. And so some of these you may have heard of. Single rings are cytosine. Sometimes we represent that by a C. So these are nitrogenous bases that are found in nucleic acids. Thymine, which we represent with a T. This is in DNA only. And then we have uracil, which is represented by a U, and this is in RNA only. I don't know if that's coming back to you from regular biology. So these are your choices of single ringed, and we're just talking about the base here. And then B, double ringed. All right, so we have adenine, which is A, and we have guanine, which is G. All right, so let me show you figure 327. So here we are. These are the nitrogenous bases, so it gives a little bit more detail. So this is your C, T, and U, single rings. This is actually showing you the atoms. And then these are your double rings. They're called purines. And so those are your A and G. And then remember, making up a nucleotide, you have your phosphate sugar base. Your sugar can change whether or not it's DNA or RNA, having deoxyribose or ribose. If you look here at DNA and RNA, why it calls it's deoxy, D means to remove, oxy is referring to oxygen, so you've removed an oxygen. If you look here, ribose has an OH right here, DNA does not, so it's missing an oxygen. That's why it's called that. So if you notice back over here, this whole thing is a nucleotide. And notice here it says nucleoside. A nucleoside is just if we're not looking at the phosphate groups. So you have a nucleoside and a nucleotide. All right, so let's go back to this. All right, so when we look at this, then we could say this whole thing right here is a nucleotide, the whole thing. That's the phosphate, the sugar, and the base, right? I didn't put the S here for sugar. All right, so then let's look at then a nucleoside is the part that is just here. So without the phosphate group. So this is a nucleoside. So if you see that word, I just wanted you to be familiar with what it is. A nucleoside is just the base and the sugar. All right, base and sugar. So the phosphate, sugar, and base. Okay, so a polynucleotide, which DNA and RNA are made up of many of these, so hooked together. So how do we hook them together? So number 3 here, dehydration reactions occur to join nucleotides together. Together to build a polypeptide. Polynucleotide, I think I said peptide. To build a polynucleotide. All right, so let's draw this. And so let's draw the nucleotide. So one nucleotide is a phosphate, and then your sugar, and then your base. Okay, so then how do we hook another nucleotide to it? One molecule is going to lose an OH, one is going to lose an H. All right, so what's going to happen is I'm going to put this bond, this is the new bond, so water would be lost. All right, here. And this new bond that is formed will then hook onto the next phosphate group of the next nucleotide. So this is one nucleotide, so then we have phosphate, sugar, and then your base, and I'm just gonna phosphate, sugar, base. All right, and you would continue on. So the next nucleotide would come in, and well, let's draw one more for good measure here. So it's going to bond here, and you're going to have your phosphate group of the next. All right, like this. All right, so then you'll have your sugar, and then you have your base. And I'm just going to draw single rings for the bases here. All right, so that's one side. So what I want to show you here is that the phosphate group is hanging off at the end of one side and it's not on the other side. This right here, if we count the number of carbon atoms here, so in the sugar, so this is the first carbon, second, third, fourth, fifth. All right, so that's the 5th carbon. Notice that the 5th carbon in this sugar is not actually part of the ring. All right, it's kind of hanging off here. So they call this the 5' end of the nucleic acid. That's because of the 5th carbon hanging here. Another way to think of it is just the phosphate group. And then down here, here's the 1, 2, 3rd carbon. They call this the 3' end of the DNA molecule, or the DNA or RNA. All right, and so that is a polynucleotide, and you can continue to add nucleotides to get your polynucleotide. Every time you hook 2 nucleotides together, a water is lost, and these are also what we could say is these are covalent bonds that's holding them together. All right, I'm only going to label it on 1. Okay, so you have, they call this part here, the phosphate and the sugar, they call that the backbone of the molecule, and what's sticking out are the bases. All right, so RNA is single-stranded, so RNA would have ribose as your sugar, and then they would have these bases, and they could be in RNA U or C for the single rings. If I were to make them double, they could be A or G. So RNA just doesn't have thymine. All right. If it's a DNA molecule, it could have C or T as the single rings here, and it would have deoxyribose, or A and G. DNA, though, this would not be a completed DNA molecule. Why? Because DNA is double-stranded. So I want to show you how that works. So let's do since it's double-stranded, there's 2 polynucleotide chains in DNA. So, and they run what are called antiparallel. And so what they found in studying the DNA molecule is that the 2 strands, the bases, are kind of held together by hydrogen bonds. So I'm going to do, hydrogen bonds are represented by ... So I'm going to do that here and say that these are hydrogen bonds. And then on the other side here, they found that if you have a single ring on one side, they found that there's a double ring on the other side. All right, so here, I'm going to go ahead and draw a double ring. All right, and so I'm just going to draw a double ring like this. All right, so that's your base. All right. And I'm going to have to finish the rest of the nucleotide. So I'm going to draw a sugar. All right. Sugar. So then we just need our phosphate group. So from the sugar, I'm going to draw my phosphate group. All right. So my base, my sugar, and my phosphate group. All right, so then over here, I'm going to draw my next base. All right, so I'm going to have another double-sided base here connected to a sugar. Base, should put sugar here, sugar and phosphate group. And so then what's going to happen is the phosphate, just like here, phosphate and the sugar bind. All right, that's your covalent bond there, just like it is over here. And I've got one nucleotide, another nucleotide, and lastly my third nucleotide. So my bases, I'm not really counting the sides. You can get the idea here. My base, my sugar, and my phosphate group. Base, sugar, and phosphate group. And ideally, whoops, it's not going to bond to the oxygen. It's going to bond to one of the carbons here. All right, and then we bond 3 nucleotides in this direction. So this is a 2 polynucleotides, hydrogen bonded in the center, and a double ring and a single ring always have to go together so that you have equal space in between here, so it's not skinnier in one part and wider in another part. And so notice another thing is the phosphate group is hanging off this one end. All right, so this is your 5' end on this side, and your third carbon, this is your 3' end. All right, they call this, we'll talk more about it later, antiparallel, all right, where they run in opposite directions. All right, and so RNA is a single strand, DNA is a double strand, but this is a polynucleotide, bonded, hydrogen bonded in the center. One other thing that you hear about when talking about nucleic acids is that there is a backbone. So the phosphates and the sugars, I'm going to do purple because I haven't used this color in this picture. And this is considered the phosphate sugar backbone. All right, so I'm just going to draw. So this and this is the phosphate sugar backbone. And so that's kind of like if you think about a ladder, all right, that's the edges of the ladder, and then the bases are the rungs, all right? But that's what they mean by the backbone of a polynucleotide. So we could say a polynucleotide has a phosphate sugar backbone with bases sticking out. Sometimes it can be completed with one polynucleotide, and sometimes it's two if we're talking about DNA. All right, so let's look at the bases. When we say that the two rings go with one ring, so there are base pairing rules, all right? So let's write about the bases. Base pairing rules. So what do we know about that? I don't know if you remember from biology. A bonds with T or U if it's in RNA, and B is C bonds with G. And if you notice here, if we go back up here, C bonds with G, single ring with double ring, single ring thymine, A and T, single ring with double ring. And then if it's RNA has U's instead of T's. So that's why there's particular rules, all right? So is the size. And let's just write one other thing. I'll switch. So I'm gonna put this as number 5 about the structure here, is that the sequence of bases A, C, T, G, U determine the order of amino acids to join to make a protein. So what does that mean? Let's go back here. So if you have if you have C, T, C, the order of those bases, A, C's, G's, C's, and G's, your cells use kind of like reading, and that determines the order of amino acids. And so DNA is always contains the codes to make proteins. All right, so that's and those codes are encoded by the order of those bases. All right, so then let's look at letter B. Let's go back to B here. So we started out with A. There are 2 types of nucleic acids. And so I want to just compare and contrast these two. All right, so we're going to make kind of like a chart to compare them. All right, so there's RNA. Now, why do they call it RNA? It stands for ribonucleic acid, RNA. And then over here, there's DNA. This is deoxyribonucleic acid, DNA. Alrighty, so let's compare them. Let's do a little chart here. How do they differ? They differ on their sugar. That's where the ribo comes from and why it's called RNA. RNA has ribose as its sugar in its nucleotide. DNA has deoxyribose. In the nucleotides, they differ in the bases. So RNA has U instead of T, so they have U, A, G, and C bases. DNA has T, A, G, and C bases. So the T and the U is different in the nucleotides. RNA, the finished product, is single-stranded. And DNA, as we drew, is double-stranded. And I'm going to put here bonded by hydrogen bonds. So let's go ahead and just draw it just to compare here. We have a nucleotide, phosphate, sugar, and a base like U, connected to another nucleotide, that's the covalent bond, phosphate, sugar, and a base like G, and so on and so forth. And that's single-stranded, all right? So you can see the sugar would be ribose, and the bases would be one of these 4 bases. I just chose 2 there. Whereas this is going to be double-stranded, phosphate, sugar, and a base like T, bonded to the next nucleotide, phosphate, sugar, and a base like C, all right? And this would be your 5' end, it's the 5 of the phosphorus, and this would be your 3' end, and this would continue on. I'm only gonna draw 2 just for comparison. And then we're gonna have the hydrogen bonds. And then this C, what is it gonna be bonded to? AG, and it's gonna be a sugar and then a phosphate group. So this is gonna be the 5' end because of that phosphate group. Okay, then the sugar is going to be connected to the phosphate group of the, so this is the bond, phosphate group of the next, so the phosphate, sugar, and then the base is going to be, to go with an T, is going to be an A. So this is the covalent bond and covalent bond, all right? And so 1 nucleotide, 2 nucleotides, appropriate bases, and this is the 3' end. So they run in opposite directions, all right? And so, you know, we would continue this way, all right, to build it. One other thing that is double-stranded for DNA, I just wanted to also put here that the shape is a double helix, that once it's double-stranded, it twists, and you probably remember that from regular biology, but I'll show you here. This picture here. So this is a double strand of DNA. The ribbon is the phosphate sugar backbone, and then the bases are sticking out, hydrogen bonded to each other. So the longer ones are the 2 rings, shorter ones are the 1 ring. A's go with T's, C's go with G's. All right, and so that's called a base pair, joined by that hydrogen bond. Oops. All right, and because of the order of bases, if you have one side of the DNA, could you tell what the other side of the DNA is? Absolutely, just like I just built this side first and we were able to figure out the other side. And because of that, DNA can replicate very well because you only need one side of the DNA. And so this is how we make more DNA through DNA replication because of those base pairing rules. All right, so those are your nucleic acids.